what you people know and what I give to you. Okay. And first of all, I'm sorry to be there in between your lunch and this my talk. Okay. So we'll be talking about DNA fingerprinting of crop varieties. So this is the structure of my presentation. So we'll be talking a little about PGR and crop varieties, DNA markers, different kinds of markers which are used in DNA fingerprinting, some of the basic molecular biology techniques that are very routinely used in the lab for DNA fingerprinting, some popular DNA fingerprinting techniques, many of you might be aware of those things. What is the global status of DNA fingerprinting of crop varieties, then with respect to the Indian crop varieties, what are the status at present, and some concerns and way forward in the area. So coming to the PGR and crop varieties. By this time, you all might be familiar with what PGR are, isn't it? So plant genetic resources encompasses everything that is on the earth related to the plants, which are beneficial to the mankind. So these plant genetic resources are the basic raw material for crop improvement. Whatever new crop varieties you are going to develop, if you don't have this plant genetic resources, you can't proceed further. And from these plant genetic resources, you can fish out various kinds of novel genes for various biotic and abiotic stresses, which you can transfer to your cultivated plants. In the present regime of transgenic crops, you can have designer crops, crops of your willingness, what idiotype, what phenotype that crop has to be. You can design as per your whims and fancies. And given the poor acceptance of these transgenic crops in the present era, plant genetic resources, they find even more valuable place today. So from heritage of mankind to the sovereign rights of the states, this is the path these plant genetic resources have traversed. Earlier, before CBD, Convention on Biological Diversity, which was signed in 1992, and India is also a signatory to that. Before that, plant genetic resources were considered as heritage of mankind, freely available to everybody. But after signing that, they became the sovereign rights of the states, means sovereign rights of the nations. Whatever is there in India, that belongs to India. Whatever is there in United States, that belongs to United States. If such is the scenario, if states have their rights on those plant genetic wealth, then protection becomes very, very important. Why should we give to other body or some other country whatever we have? Why don't we harness benefit out of that, isn't it? So protection of those plant genetic resources is very, very important. So DNA fingerprinting is one technique which can facilitate that protection. So we'll be discussing a lot about this. So these plant genetic resources, as you know, that it encompasses everything, and it is a very small part, and crop varieties are a very small part of that whole chain. This is the biodiversity, whole gamut of biodiversity. From that biodiversity, this agrobiodiversity is a small component. PGR is further down a component of that whole agrobiodiversity, and crop varieties are just one part of the PGR. So these plant genetic resources consist of everything: land races, farmers' varieties, parental lines of hybrids, released varieties, wild and weedy species, primitive cultivars, genetic stocks, and I have added even of late these genetically modified organisms or living modified organisms. So before we proceed further, it is very important to define a variety. How can one define a variety? Can you define a variety? What is a variety? Anybody?
Cultivars which are notified in Gazette of India, having one distinct trait.
Okay. So these have different connotations. From farmer's point of view, variety is different. From botanical angle, variety is different. Okay. So we can define varieties as subdivision of a kind identifiable by growth, yield, plant fruit, seed, or other characteristics. This is This definition as per the Seeds Act of India 1966. International Code of Nomenclature for Cultivated Plants. This defines an assemblage of cultivated plants which is clearly distinguished by any characters. That those characters can be morphological, physiological, cytological, chemical, or others, and which, when reproduced, retains its original distinguishing features. Okay. This is very important to know, because I will be talking subsequently some other things also. UPOV, International Union for Protection of Plant Varieties, defines a variety as a plant grouping within a single botanical taxon. Plant grouping within a single botanical taxon, within a single species, you can say. The lowest known rank, which grouping, irrespective of whether the conditions of the grant of breeders' rights are fully met, can be. Whatever grouping you are assigning, that grouping, which grouping is defined by the expression of the characters resulting from a given genotype or combination of genotypes. That is one thing. Or that grouping, whatever way you are grouping that, that is distinguished from any other plant grouping by the expression of at least one of the said characteristics, and is considered as a unit with regard to its suitability for being propagated unchanged. So that characteristic, when it propagates to the next generation, it doesn't change. Okay. Essentially derived variety. Have you ever heard of it?
Yes, sir.
Okay, great. A variety predominantly derived from another original variety, initial variety, which retains the expression of the essential characteristics from the genotype or combination of genotypes of the initial variety. This is again a definition by UPOV. So when we are talking about the DNA fingerprint, we will be talking about DNA fingerprint techniques and other things. So it becomes very, very important to distinguish a variety from essentially derived varieties. For example, somebody develops a variety over a period of 20 years, 15 years, 10 years. I simply make some cosmetic changes in that variety and say that variety is mine. Okay. And 99.99% both the varieties are similar. How do we distinguish them? So protection of crop varieties, how can we protect those varieties? There are various methods. One, we are employing at NBPGR also registration of the varieties or germplasm. In somebody's name or somebody's community, we can register that. UPOV and our Indian system, that Protection of Plant Varieties and Farmers' Rights Authority, wherein we are using these parameters: distinctness, uniformity, and stability, which is a statutory requirement for a variety to be called distinct. And that variety, whatever variety is there in question, that has to be novel, distinct from the existing varieties, uniform and stable in its essential characteristics. And other methods of protection are granting of plant breeders' rights. But in India, we have a very unique system, that is Protection of Plant Varieties and Farmers' Rights Act. Okay. So these are different types of IPRs. I'm not going into the details of these. But we have a legal instrument in our country for providing protection to the plant varieties. We have legislations in place: the Patents Act 1970, the Biological Diversity Act 2002. But as far as protection of varieties is concerned, that is the Protection of Plant Varieties and Farmers' Rights Act 2001. Under the purview of this act, everything related to protection of those varieties come into place. So why do we need DNA fingerprinting? As I told you earlier also, for example, new varieties are often derived from existing protected varieties. I make small cosmetic changes and say that variety is mine, isn't it? Is there a system to check those things? So that is the place where DNA fingerprinting comes into picture. Hybrids. Many times people are developing new hybrids. They're not disclosing the parentage. What are the parents involved in the development of that hybrid? Okay. So hybrids are reproduced illegally without a license from the original developer. I developed, say, for example, a hybrid in maize. Somebody stole those parental lines, and he can reconstitute that hybrid from those parental lines. Okay. So protection of those parental lines also becomes very, very important. So that is the place where DNA fingerprinting comes into picture. We are regularly growing those for identity testing or purity testing, grow-out tests. Have you heard of these things? Grow-out tests. We are conducting the field. We are taking morphological observations for maintaining the purity of a particular variety. So can we replace those grow-out tests? Because grow-out tests means growing in the field, letting the plants go to the reproductive phase, harvesting seeds, and then analyzing. Why not we take out only a few seeds or samples, leaf tissue, small leaf tissue, analyze DNA, say whether that is pure or not. So can we think of replacing those field trials with the grow-out tests? Sorry, replacing those grow-out tests or field trials with the cheaper techniques. Okay, and these DNA fingerprinting techniques are feasible on all kinds of material, be it is the living plant material, tissue, leaf, bud, flower, stem, whatever it is, or it can be dead even. Herbarium specimens, you can maybe 100 years ago somebody has put some samples in the herbaria. You can just take out those leaf tissues and extract DNA, and you can just see the profiling pattern. So I'll be talking about little about the DNA markers and DNA fingerprinting. What is a marker? For example, how many students are there? 25, 30? If I say that I want to distinguish each and every one of you, I may say that based on the gender, I can say 15 are males, 10 are females. That is one character, isn't it? Or I can say that there are, what can I say? There are guys who are wearing specs. 15 guys are there who are wearing specs, and 10 guys who are not wearing specs. That is another feature to distinguish them. Okay. So we can think of like that many characters. So that, for example, wearing a specs, that is a marker, isn't it? Or eye color, based on eye color, or stature, height, we can say that. Okay. Based on that, those traits we can distinguish, and I can classify. And I can keep on adding traits until or unless each and every individual is distinct from the other. Okay. Likewise, and we are making use of the markers for identity testing, for identification. So what is a marker? Any genetic element. The examples I gave, those are just physical features. Those wearing a spec, that's just to make you understand. That may not be, that's not genetically inherited. Okay. So marker is, I think there's a problem with this. उसको निकाल दो, इसको लगा दो. ठीक है, it's okay. It's okay. Any genetic element. That genetic element can be a locus, allele, DNA sequence, DNA fragment, chromosome feature, which can be readily detected by phenotypic, cytological, molecular techniques. Okay. For example, in tomato you can see skin color, or maybe allozymes, proteins, or cytological features. For example, your chromosomal knobs in case of maize. It can be anything. And the basic requirement is that marker has to be genetically heritable. That has to be genetically heritable. It should follow Mendelian inheritance. Otherwise, it's not a marker. If you can't track that marker through genealogy, then that means it's not heritable and it's not acting as a marker. So an ideal marker should be, whatever marker for identification we are using, that should be highly polymorphic, reproducible. Once I repeat those experiments, that marker should give the same results. Codominant marker, preferably should be codominant. Evenly distributed in the genome. It should be discriminating. It should not be subjected to environmental influences. It should be neutral, not subjected to selective sweeps. It should be inexpensive, and it should be easy to measure. So these are some of the features which make which make a marker to be an ideal marker. If we will be talking about different kinds of markers, so we can see which marker fits under which category. Okay. And DNA markers, there are large number of markers. Virtually speaking, infinite kind of markers you can generate using DNA. I talked about the physiological features, specs, high color, height. So these are limited markers. For example, I have to distinguish in this group 25 individuals, maybe I have to distinguish 500 individuals. So these markers will be becoming limiting. I don't have sufficient markers to identify. So DNA, through DNA you can generate infinite number of markers. For example, you take a sequence which is 10 nucleotides long, okay, and at each base we can think of replacing with 4 nucleotides, substituting with 4 nucleotides, ATGC, isn't it? So, for example, one variant of this sequence is where this A is replaced with T. Another variant is this T is replaced with C. Another variant is this G is replaced with A. So, if you have a 10 nucleotide long sequence and each base can have 4 different options. So, this sequence can have 4 raised to power 10 possible sequence arrangements. Okay. So, this number is unimaginable, and human genome is, as you all know, 3 billion base pairs. 3 billion base pairs, 4 raised to power 3 billion. You can just imagine, which is, you can't even write down that whole sequence. That many number of variants you can create. So that is why DNA markers are very, very useful for identity testing, because number one, they are unlimited in number, and secondly, no influence of plant development. When you are growing those plants, you can have your DNA from stem, leaf, flower, bud, any part. DNA is same. We are looking at DNA. What is DNA? Deoxyribonucleic acid. We are looking at physical entity, something physical. Okay. So there's no influence of plant development on the DNA. No secondary pleiotropic effects. No influence of environment. No epistatic effects. So because DNA is a physical entity, we are looking variation in that DNA only. It's not the expressed products of DNA, where we can anticipate some variations. So these DNA markers I have just listed many applications. For example, can be used in genetic studies. This can be used as breeding tools, production of in quality assurance or quality control, legal applications. And under each category, we have large number of applications. I'm not going to dwell on these. And under legal applications, we are focusing on this plant variety protection. Only one aspect. So DNA fingerprinting is what essentially it is. It is a way of identifying a specific individual rather than simply identifying a species or some particular trait. Many times people confuse DNA fingerprinting, DNA barcoding, DNA genotyping, okay, profiling. So DNA barcoding, there's a lot of difference between all these terminologies. Within a species, identifying cultivars, that is DNA fingerprinting. A level up, species delineation, that is DNA barcoding. Okay. If you dive into the literature, you'll find that DNA fingerprinting is also known by other names as DNA typing, genetic fingerprinting, DNA profiling, but many other names. But there still are, between all these terms, there are still small variations. For example, you'll find DNA fingerprinting and DNA profiling are synonymous in the literature, but it is not true. Profiling is very preliminary. Fingerprinting, when you have statistical authenticity of those profiles, only then you'll call it fingerprinting. Okay. And as a technology, this DNA fingerprinting has been around us since 1985, when first of all, Sir Alec Jeffrey, he invented it. And in humans particularly, it is used for paternity testing or maternity testing or criminal identification, and there are numerous applications in plants as well. Fingerprints versus DNA fingerprints. We have DNA fingerprints. Okay. and these DNA fingerprints are unique to each individual. You might have heard that no two individuals have similar DNA, similar fingerprints, thumb impressions. Okay. So DNA fingerprints are unique to each individual. But the difference is that the probability of two unrelated individuals having identical DNA fingerprints is much lower than that of their fingerprints. I pick up two random individuals, and when I say that what is the probability of them being identical. If you use DNA profiling techniques, that probability is very low. That means its detection power is very high, and as compared to the fingerprints. Yeah, this is very important. Fingerprinting in humans and plants. We shouldn't get carried away by saying that, in other words, it is very complicated in plants as compared to the humans, because in humans we are dealing with only one species, Homo sapiens, and there are a couple of races which are also not that 99.99% they are similar. But in plants, it is, you touch upon any plant, for example, cotton plant, you will find lot many species, Gossypium hirsutum, Gossypium barbadens, Gossypium arboreum, herbaceum, Raimondii, so many species. Each species is different, and within each species you will find lot of varieties. Okay. So you need to develop DNA fingerprinting protocols for each species. Likewise, we have so many plants, so many cultivated plants. And fingerprinting, many times people keep coming to our labs and they say, what is new you people are doing? But it is not simple as is in humans, because we are there in humans we are dealing with only one species, Homo sapiens. But similar protocols as we have in the humans, we have to develop for each and every crop. So that is one species only. Multitude of species, subspecies, races in plants. One species, similarity in requirements. Whatever DNA fingerprint profiling you want to do, similar set of equipment, similar kind of infrastructure you need. But we have here different diverse requirements. Humans all outbreeding, whereas in plants you will find that some plants are vegetatively propagated, some are crossbred, outcrossing, self-pollination, every kind of breeding behavior you will find in the plants. In humans, one individual, one profile, but in plants we can't say that. A variety is not an individual. It is group of individuals, it's a population. Okay. So in humans, one individual, one profile, but in varieties, different compositions. We have pure lines, we have inbreds, we have open pollinated varieties, we have synthetics, we have composites. All sort of range of breeding behavior in plants are there. Subtle substructures means there's grouping and some subgrouping in humans that is not that much distinct, but in plants, strong to weak substructures. Allelic specificity in races, common alleles across groups. Applications in humans include identity, paternity, criminality testing, all these things. But here we have identity testing, seed purity testing, relatedness, genetic makeup. So many other things are there. So the various. In a nutshell, what I mean to say is it is much more complicated than what we usually call in human beings. Coming to some of the basic techniques that molecular biology techniques, then we will be coming to the techniques of DNA fingerprinting. Many of you are familiar with this, just as a repetition. So PCR, electrophoresis, these are the two very common techniques used here. PCR was first of all introduced by Karimalis in 1985, okay, and it is just exponential multiplication of your DNA fragments. So this is just a schematic presentation, denaturation, annealing, extension. PCR you might have done, all might have done. So I'm not going to into the details of this. Gel electrophoresis, once you carry out PCR, it is separation of macromolecules, be it DNA, proteins, RNA, based on size and charge. We separate those macromolecules. Okay. Since DNA is negatively charged, it will move towards positive end. So we can separate those fragments. So longer fragments will move shorter distance, shorter fragments will move longer distance. So, based on that variation in the fragment sizes, we can identify individuals. So, this is just another continuation of the gel electrophoresis. Coming to some of the basic techniques where these molecular biology techniques are being used. Some popular DNA fingerprinting techniques. If you dive into the literature, you will find large number of techniques which are modifications of some of the basic techniques. Okay, for example, this is a very, very long list, and I have probably compiled up to 2010. Restriction fragment length polymorphism, one technique, variable number of tandem repeats, allele specific oligonucleotides, oligonucleotide polymorphism, ASPCR, SSAP, STS, STMS, I am not going to RAPD, this is very, very common technique. Okay. Simple sequence repeats, CAPS, DOP, PCR, MAP, SCAR marker, SNPs, sample, ISSR. So, this list continues. So, this is again continuation. So, these all techniques, there is nothing much complicated in these all, just modifications of some of the basic techniques which we will be dealing here. For example, restriction fragment length polymorphism. This was the very common technique used in the humans first of all. Restriction, as the name indicates, we digest the DNA first, and after that digestion, whatever the fragments of different sizes we get, those are electrophoresed, and based on the differences in the size of those fragments, polymorphisms are detected. And in this technique, we make use of probes. That is very, very important. Those probes are to be designed in a very specific way. And those probes, for example, in human identification, those probes are from snakes. Okay, those are used from snakes. So, in RFLP, the basic thing is the use of enzymes. Okay, restriction enzymes. These are very, very restriction endonucleases are used, which are of different types. They can be producing or staggered ends, or they can produce blunt ends. So, based on that usage, as I told you that using DNA fingerprinting, you can generate markers infinite virtually. For example, if you are using this enzyme, your fingerprinting patterns will be different. You have lot many options to use enzymes. Using this, the patterns will be different. Using this, pattern will be different. So, for example, this generates some profiles. And we have around thousands of endonucleases. So, you can use endonuclease of your choice and produce variations in the DNA. So, how do we classify organisms in distinguish varieties using RFLP? For example, you have two varieties, 1, 2. You extract DNA, cut that DNA with certain restriction endonuclease of your choice. For example, this is the DNA of genotype first, variety second. So, this is the recognition site. It will cut here, 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 here. You see here in variety first, this site G, this is mutated. Here it is GAAA, here it is GAG. So, this enzyme recognizes a sequence which is GAA-TTC. When it is recognizing that sequence, it will cleave it here, but it won't be able to cleave it here, isn't it? So, what will happen is you will get here in genotype first, variety first, a fragment which is very long from here to here, because here it is not able to cut it, and here is shorter fragment. You carry out electrophoresis and you will visualize that fragments are differential in their mobility. So, and then you carry hybridization with the probes and southern hybridization and visualize those fragments. So, you can distinguish using this your variety first from the variety second. RAPD, random amplified polymorphic DNA, is a very, very basic technique propounded in 1990s by Sir William McClintock, and parallelly by another workers in the 1990 itself. There were two papers. It is a very simple technique. As in many of the techniques, we use two primers, forward primer and reverse primers. Here we use only single primer, that is the difference, which acts as forward primer as well as a reverse primer. Okay. Single species of primer is used here. And after that, we carry out PCR, we sort out the fragments using electrophoresis, and this is how a typical gel image looks like using your RAPD profiling. So, how to interpret those gels for fingerprinting? Probably I will be needing another lecture. Data analysis, I won't be developing here because that will take at least 2-3 lectures altogether. So, I have made my presentation very, very basic. So, this is how this technique RAPD works. We put in a vial all things required for PCR to be carried out: primer, buffer, water, MgCl2, DNTPs, your template DNA, and carry out PCR and amplify and resolve through agrogel electrophoresis. Another technique, AFLP, amplified fragment length polymorphism. It is a combination of your restriction based and PCR. Okay, both methodologies are used here. Okay, and this is how typical gel, AFLP gel looks like. This is how this procedure works. We use 2 restriction endonucleases here. One is rare cutter, one is frequent cutter. 2 enzymes are used. Okay, for example, these 2 enzymes are used. We produce fragments, and selectively those fragments are identified who are having at one end the rare cutter flanking end and on the other the frequent cutter. So, we ligate adapters to these ends. So, these are adapters are ligated. We design primers, and after designing primers we carry out PCR in 2 different steps: selective PCR amplification, pre-selective PCR amplification, and selective amplification to reduce the complexity of the genome. And we carry out PCR, and then we resolve through your denaturing page, because you generate large number of fragments unlike your RAPDs. So, I am not going into the details of all these how these polymorphisms are generated, otherwise it will take another 10 minutes to explain this slide. SSRs, simple sequence repeats. This is another technique. In all the genomes, be it plants or the humans, some of our nucleotides, they are repeated. Okay. For example, A nucleotide is repeated, it can be repeated n number of times. So, this is mononucleotide. Or dinucleotide, GT is repeat motif. GT can be repeated n number of times. Here it is 6 times. Trinucleotide repeats. CTG, it can be repeated CTG. That is the beauty of all the genomes. So, you will find a lot of repetitive sequences. For example, DNA fingerprinting that we carry out in humans, variable number of tandem repeats. Those are big chunks repeated. But in SSRs or microsatellites, small nucleotide motifs are repeated. Mononucleotide, di, tri, tetra, these are repeated. And what interesting feature is that when these are repeated in the genomes, they are flanking sequences. For example, GT, this is repeated 6 times. On the upstream of this and downstream of this, those are conserved in nature. So, those have been exploited to use these sequences as this technique as DNA fingerprinting technique. Because when this upstream and downstream sequences are conserved in the genome within species with respect to the SSRs, not above the species level. So, we can use them as markers for designing our for distinguishing our varieties. Say for example, you have variety 1, variety A, variety B. So, this AT repeat motif is repeated 8 times in variety A, in variety B it is repeated 10 times, and this is conserved, this region. This is same in variety A, variety B, variety C, whatever variety you take. Okay. So, this is conserved. So, we can design primers with respect to these 2 sequences. This is 5 prime, 1 primer, this is another prime, forward primer, reverse primer. So, you carry out PCR, AT, this is repeated 8 times. So, we expect 60 nucleotide long fragment. Here we expect 20 nucleotide long fragment. So, this fragment will move shorter distance compared to this fragment. So, we can distinguish these 2 varieties. Another marker technique of very latest utility is SNP, single nucleotide polymorphisms. Single nucleotide from SNP marker, these are defined as a single base change in a DNA sequence that occurs in a significant proportion, more than 1% in the population. Okay. So, for example, you will find these SNPs to the tune of 1 SNP per 268 base pairs in rice. Likewise, 1 SNP in per 280 base pairs. So, you can produce a large number of markers, large number of SNPs, and there are a lot of discussion going on whether they are useful for fingerprinting per se, but I am not of the opinion at the moment whether these are useful for fingerprinting. There are many reasons for that. Many people are advocating that these can be used. So, we have different methods for detecting these SNP markers. I am not going into the details of these. That is one of the reasons for not using these, because we have different number of platforms for identifying those SNPs. There is no harmony for using which method to use for identifying those SNPs as compared to the SSR markers. SSR markers, I think they are the most robust types. So, these are different methods, genotyping platforms for assaying these SNPs. So, all these markers can be classified as first generation markers, second generation markers, third generation markers. As I showed you a very long list of those markers, techniques. First generation means RFLPs, RAPDs, extensive, most commonly when the hybridization based markers were in vogue in 1980s, 90s. Then PCR based markers, then sequence based markers. There are many ways of classifying these markers. For example, these can further be classified as random DNA markers, wherein we don't know or we can't track in the genome where from that marker is coming. Or these can be gene targeted markers, or these can be functional markers from the domains of genes which are responsible for certain function. You can make a comparison for fingerprinting, which marker is used. I have made a comparison of only 3-4 techniques, otherwise I have that long table of comparing all those techniques also. RFLP, SSR, RAPD, FLP, these all markers are compared, means which marker is better for what are the requirements. For example, dominance, codominant markers we always prefer. So, these are all, for example, RFLP, AFLP, RAPDs, these are dominant markers, RFLPs, SSR, codominant markers. Each and every markers have their pluses and minuses. I am not going into the details of those. So, coming to some of the developments at the global level. You might have heard of this UPOV. Have you heard of it? International Union for Protection of Plant Varieties. UPOV, there are many member countries of UPOV, they don't recognize DNA fingerprinting technology to be used for identification. They are more relying on DUS, distinctness, uniformity and stability testing for identity. Okay. But they say that DNA markers or DNA fingerprint techniques can be used as supportive evidence. Okay. DNA profile is not the basis for obtaining the protection of a variety, although this information may be used as supportive information. And what is the reason they don't argue for these? Because they say that sometimes what happens, two varieties are there. They are phenotypically different, but genetically they can be same. Or the other way around, they are genetically different, but phenotypically they can be same. Okay. And further they say that it might be possible to use a limitless number of markers to find differences between varieties at the genetic level that are not reflected in phenotypic characteristics. DUS we are using mostly for characterization, not for identity testing. DUS we can do away with unless we have whatever characters are used for DUS. We have one-to-one linked markers, molecular markers linked with those traits. Only then we can say that okay, we need not to grow those varieties in the field and record those morphological observations, because our DNA markers, they are exactly one-to-one linked with the traits of interest. So we can substitute these, but that is not the case as of yet. That is what they say, that molecular markers can be used only as a method for examining dust characteristics if there is a reliable link between the marker and the characteristic. Because markers are different, and morphological characters are different. They are governed by genes which are located at different positions. Your markers are located at positions which are totally different, not linked with the gene positions for those characters being used for characterization. Further, they say that genetic distances or morphological distances, a combination of phenotypic distances, you calculate phenotypic distances, and molecular distances or the DNA based test distances, to improve the selection of varieties to compare in the growing trials if the molecular distances are sufficiently related to phenotypic differences. If your molecular distances are related with the phenotypic distances, then it can be thought of as a substitute, but those are not. Many times we do many experiments in the lab, molecular distances are totally different. They are not correlated with your phenotypic distances at all. So, for example, in some of the countries, this is the status. UPOV has this working group on biochemical and molecular techniques, and particularly for DNA profiling. There are many countries, and they are moving ahead with many of these advancements in DNA profiling of the varieties. In France, for example, these are very, very pilot studies, not in-depth studies. They are advocating the use of SNP markers. For example, in South Korea also, they say that SSR will be replacing the SNPs. SNPs will be replacing your SSR markers. USA, there are many countries, they are coming up with the chips for identity testing of the cultivars in some selected crops. UPOV is one such organization working at the global level. We have many other organizations working on these markers for identity testing. For example, another is Organization for Economic Cooperation and Development, International Seed Testing Association, Association of Official Seed Analysts. Now, of late, for the last 3-4 years, these all associations are coming together and harmonizing things, so that we have unified markers, unified protocols globally to address identity testing of varieties. I forgot to tell you, for example, in humans, we are using 13 SSR or STR markers globally. Okay. In the United States, out of those 13, 9 STR markers are used for identifying all those human populations of the United States. In India, we are using I think 20. And after so many years of research, they have harmonized everything in one species, as I was telling, and come out with 13 STR markers for distinguishing all the population of the world. But we don't have those unified protocols, unified markers. You touch upon, for example, wheat. We have different set of markers in India. Somebody else is using another set of markers. So we don't have any unified unanimity in the usage. So many of these associations now are coming together to harmonize these kind of things. In India, we have done a lot of work for the last 22 years. We have been working on this area, and one requirement is there by Central Subcommittee on Crop Standards Notification and Release of Varieties. Once a plant breeder develops a new variety, that variety has to be DNA profiled, and there's a long proforma. That proforma has to be filled up by those plant breeders, and section 5 of that proforma says this: parentage, with details of its pedigree, including source from which variety inbred A, B, R lines of hybrid has been developed, source of material in case of introduction, and this is DNA profile of variety hybrid inbred A, B, R lines of hybrid vis-a-vis check variety or line. So this is the requirement at the ICAR level. So this institute here at NBPGR, we are particularly nationwide addressing these two issues. One is this, and one is this, providing identity to the material, IC numbers. Whatever new variety is developed, whatever germplasm is there, all those materials come to us, and we are providing identity. That is one thing. And whatever new variety is being propagated or identified or released, notified, seed lot comes to us, to our lab, and we carry out DNA profiling of that. And under that, for example, this DNA profile, what that performer says, they ask those breeders for information on the molecular discrimination should be provided. Such information can be developed at crop-based institutes or NBPGR or other labs. The information should include details of amplicons, name, sequence number, primer sequence with reference to polymorphic markers. The relevant photographs should also be attached. It's a very, very vague statement, and nothing is clear in this. Means, and we are developing those protocols for making it more clearer. Okay. For example, genetically engineering approval committee, appraisal committee, that is insisting. We are developing a lot of transgenics nowadays, and this committee is insisting on DNA evidence to prove background genotype of transgenics for the last 10 years. Whatever transgenic material you are developing, what is the background material you used? In which background you transferred that transgene? So that background material that needs to be fingerprinted. Why? Because many times transgenics, that gene of interest is transferred in a background, and a person X is saying that this is variety this. Same genetic background, slight modification. So in the same genetic background, many people are parallelly attempting to transfer that transgene. So what is happening? The genetic base is narrowing down, and that is vulnerable to various biotic stresses. So genetic base has to be very, very variable. So for that into consideration, taking that into consideration, this is a requirement, just to infer whether the genetic background is diverse or not. So we have made this, as I told you in the previous slide, fingerprinting essential for release of crop varieties. So whatever variety is to be released now, until or unless those profiles are attached, which we provide from our lab, so it won't be released. So this is some of the development in our labs here. Protocols have been developed in using different marker techniques in 30 different crops of national importance. Okay. Molecular profiling of varieties, land races, parental lines of hybrids, and registered material of crops such as rice, maize, sorghum, cotton, pearl, millet. This is listed. And apart from that, we have many other things also. And we are rendering this DNA fingerprinting service to various public and private sector organizations or institutes. And many times in our lab, many samples come which are confiscated from customs department. Sometimes samples are smuggled. They're coming through customs departments to identify, for example, last year only, almond, some black pepper samples, some maize samples which were imported through Nepal into India, sometimes samples through Sri Lanka, sometimes through Pakistan. They're coming, and they are being identified whether they are really of Indian origin or not, or they are of some foreign origin. So like those questions are being addressed. So for example, this is just during the last 4 to 5 years, 13 to 18, profiling. For example, we have carried out in 640 different plant varieties of 38 crops, and this is the list of those crops which we undertook: almond, areca nut, bay leaf, bitter gourd, chickpea, cluster bean, cotton, cowpea. So this is a very long list. Keeping in mind, for example, as I told you that there are no harmonized protocols. In previous slides, so that NBPGR can do DNA fingerprinting, or some other crop-based institutes can also do, but there is no harmonization in the protocols. So DG last year, the Director General ICAR made a committee of 10 people to harmonize those protocols, and we are very instrumental in that. We have been working on that day and night, and this committee was to discuss and decide scientific, technical, and legal issues related to DNA fingerprinting of crop varieties, to identify crop-based experts to finalize logistics and protocol development in each identified crop, and to finalize the modalities of operation of centralized service involving major crop-based institutes. And this will be the outcome. We had 5 meetings so far. Last meeting was held in April, I think, and the outcome of this was we were to formulate a big project, and we have formulated that project. We have identified crops, 20 different crops. 5 crops were added later on of horticulture crops, 25 crops are added. So we are harmonizing those protocols in those 25 crops right now. So once that protocol is developed in each of the identified crops in those 25 crops, that will be made public. That will be submitted to DSE for approval. If once that is approved, that will be made available to everybody in the country, and they can use that protocol to profile or fingerprint their own material. And this we are anticipating network project to do this all groundwork. So for example, let us say any crop, maybe cotton or maize, wheat, we have identified nearly 200, 500 different markers. Those markers will be given to the identified labs. So they'll be validating those markers, and once those markers are validated, once those are found and validated, handful of markers, 10, 15, whatever those are, which are working across the labs uniformly, those will be identified, and a protocol will be developed. Everything will be very well written down. What is the DNA extraction procedure? Which method is to be used? What is the electrophoresis procedure? How many micrograms of DNA? Primer, PCR, which company? Everything has to be very detailed protocol. Which markers? What is the forward sequence? What is the reverse sequence? Which electrophoretic technique? Whether it is capillary electrophoresis or agarose based? So all the steps will be defined so that any layman, if starts working on that, he can do that. So and NBPGR will be the nodal organization for that, and we have identified 11 different pioneer institutes in the country. So we will be working in network with those, and we will be coordinating that activity. So some of the concerns, as many of the things I have already discussed, scientific issues related to this, development of quality standards and monitoring protocols. That is very, very important. As I told you, that protocol development, everybody is using protocols as per their whims and fancies. So we need to have quality standards. And which mark, for example, we take example of wheat. You are using, say, for example, 25 markers for DNA fingerprinting. Why 25 only? What is the criteria? Why at that number only, 25? And why those 25 only? What is the criteria for selecting those markers? So everything, we have done that groundwork, okay, based on many parameters. So these things need to be extended to many other crops also. Selection of fingerprinting marker system. Which system? SNPs or SSRs, or what technique for a particular crop? Based on reproducibility, discrimination power, and possibility of robust database. Databasing, I'm not talking, otherwise that is altogether a different topic. And for example, we have many crops, native crops of Indian origin, where we don't have markers, SNPs or SSR markers. So we need to develop those first, then those can be applied for fingerprinting or cultivar identification. So that will again require a lot of groundwork for those crops where those markers are not available. Development of plant DNA fingerprinting database at the national level. Once we have identified those 20-25 crops, we need to develop a database first. For example, in our country, for example, say we have 1,000 varieties of rice. Do we have DNA profiles of all those 1,000 varieties available, so that when a new variety comes, we make a match with all those 1,000 and can say whether it is similar or different. If it is different, that only, then only it will be released. If it is same, what is the use of releasing that as a new variety? So for all those crops, as I was telling, that it is very simple in humans. Many of the countries have developed so many databases, but in crops, none. Okay, and we have so many crops. Many times people argue that it is so simple, but in humans in India, for example, CCMB is working, and there are 11 different technical people, so many scientists working on one species only, and here in our labs only 2-3 people are working for all the crops. You can just imagine. So standard reporting format for DNA fingerprints. Somebody is reporting fingerprints in their own way. Somebody else is reporting in their own way. Do we have a unified standard protocol, standard procedure, how it has to be reported, how it has to be put in the database? So standard reporting format has to be there. Some organizational issues. for example, we need to establish an advisory body at the national level and monitor that advisory body, that advisory body should also look into various quality control parameters. When we are developing markers and different labs are working, some people may be very casually reporting. So somebody needs to control them, check their profiles, check their labs, check their data, whether that is authenticated or not. Only then we need to identify a referral lab for DNA finger at the national level, have a network of different labs. And this network, not only for DNA fingerprint or crop varieties, network for crop specific, wheat DNA fingerprinting network, rice DNA fingerprinting network. It has to be like that only. Above all, we need to create a legal awareness. As I told you that many times people get confused what DNA fingerprinting, profiling, typing, everything is same for everybody. That's not the case. DNA fingerprinting is very, very authentic because profiling, you just take a marker, carry out, take a few samples, generate profile, some bands, black and white bands, that's a profiling. But what is the allele frequency, particular band? What is the allele frequency of that band in the population? There are lot many population parameters you have to take into consideration. Lot of statistics involved to call a profile to be a fingerprint, to qualify that profile as a fingerprint. It's not easy. So we need to develop a national database also. Using common set of markers, we need to have a allele frequencies database, and we need to decide whether that database has to have open access to everybody, or what are the modalities for that for the usage of that database. So many things are to be done. So ICAR Institute, of course, NBPGR has taken a lead role in this. So just comparing plant DNA fingerprinting is the human DNA profiling bill was conceptualized in 2007, and its drafting process began in 2012. And this bill says that there has to be a national DNA data bank and DNA profiling board. Okay, so this bill and that board will define the standards and controls for DNA profiling. In humans, I'm talking about certified labs that will identify everything that will look into. In 2016, Department of Biotechnology forwarded a draft bill, the use and regulation of DNA-based technology in civil and criminal proceedings, identification of missing persons and human remains, Bill 2016. And in 2017, it was revised as the DNA-based technology, Bill 2017. And it was likely to be tabled in the Parliament during 2018, but it was later on said that it was argued that it should be referred to a standing committee to resolve existing problems first. So it hasn't been into implementation stage. That is far that will take a lot of time. So it is just a proposed legislation in India. So you can just imagine what would be the status of plant DNA fingerprinting bill, because science and technology is so much advanced in humans, and we are talking about profiling bill in plants. So it will take, I think, a lot of time yet. So we have many other countries, for example, USA. They have DNA Identification Act 1994. Other countries, UK, other countries, Norway, Finland, Belgium, well-defined system is in place for human identification. And we are just contemplating these things in human at the moment. So it is a long journey for us. Thank you very much for your patient listening. Any questions, clarifications?